Hello everyone, this is the Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And this is the live session 2. And we have so far what we have covered in this course. So in the live session 1, I have covered until the module 4. And in the module 5 to 8, we have covered about the overexpression of the proteins. We have discussed about the recombinant DNA technology and so on. So if you have any questions related to this particular topic, you can raise your hand and you can ask me the queries. Alternatively, we can also have the one-to-one interaction. So in that case, Shusti, do you have any question?
No, sir.
Okay, so what's your background right now?
So I'm studying engineering, first year, sir. We have right now.
Can you repeat? I can't hear you. Padmasri, do you have any question?
No, sir.
Okay, please ask me. What is the background, Padmasri? Padmasri? So make a just to make the interaction more fruitful, maybe I should share my screen and I should summarize you what we have discussed so far, so that it will help you to to ask the questions. So I'm sure my screen is visible now, right? So what we have done, okay, in this course is we have first in the beginning we have discussed about how to identify the gene, and once you identify the gene, we can actually be able to amplify the gene by a technique which is called as PCR, or the polymerase chain reaction. And while we were discussing about the PCR, we have discussed about the mechanism of PCR, And we have also discussed about the technical details. We have discussed like how to prepare the primers, how you can be able to troubleshoot the different aspects related to primers and all that. So once you got the amplified gene, and these amplified gene are actually going to have the uh, restriction enzymes on both the sides. So you're going to have a gene which is actually having the restriction site on both the ends. Okay, so that restriction enzyme will help you in terms of so once you digest this gene with restriction enzymes, it is actually going to generate the sticky ends. Okay, so once you generate the sticky ends, right? So this is what you're going to generate. You can actually be able to generate the vector also with the similar kind of sticky ends. And when you put them together, you can be able to, you can be able to get the clone. So in the clone, you are going to have your gene, which is inserted within the multiple cloning site. So this is actually the clone what you are going to get. And then this clone you can be able to transform into the suitable vector. I think in this particular course or this particular modules, we have discussed about the transformation or the entry of the DNA into host. So when we say about the host, host could be anything. Host could be E. coli. Host could be yeast cells. Host could be insect cells, and host could be the mammalian cells. Okay. So if you have use any of these hosts, the entry of the DNA is going to be different. The method what you're going to use, you're going to do the transformation for the E. coli cells. You're going to do the transfection for the mammalian cells and as well as the insect cells. For each also you're going to do the transformation. Now once you do the transformation, you're going to get the recombinant organism, right? You're going to get the recombinant organisms, which is going to harbor the gene of your interest. And then what you're going to do is you're going to do the induction, and with the help of, so in depending on which host you are going to use, you're going to use that kind of induction system. For example, if you use the IPTG based induction system, you're going to use the IPTG for induction, and then it is going to give you a protein of your interest, plus it is actually going to have the cell lysate proteins, which is there. Okay, so after this stage, you are going to do the purification. Okay, so you're going to use the different chromatography techniques and different chromatography techniques, what you're going to do, and that's how you're going to get the purified product, and that purified product can be used for different types of biotechnology applications. Now, as far as the chromatography is concerned, the protein is actually giving us the flexibility of using the several properties which of the protein which you can actually be able to use for chromatography. You can use the charge as a property. You can use the hydrophobic pores or hydrophobic patches as a property. You can use the surface area as a property, or you can use the affinity as a property, or antigenic sites as a property. So if you use the charge, the chromatography technique is called as ion exchange chromatography. If you use the hydrophobic, then the chromatography is called as HIC. If you exploit the gel filtration surface, then it is going to called as gel filtration chromatography. And if you use the affinity, then it is going to be called as affinity chromatography. And while we were discussing about the affinity chromatography, discussed about how you can be able to generate the receptor, how you can be able to produce the ligand, and so on. And that's how we have discussed that how you can be able to prepare our affinity column and how you can be able to perform the chromatography. Now, once you produce this protein, okay, this protein has to be characterized under the different biotechnology, different biophysical techniques. So that anyway we are going to discuss in the next live session. So this is just, this kind of discussion is just to make it, you know. So this is all about the summary of the different modules what we have discussed so far. If you have any questions, you can ask me now. So we have a couple of students now, and I think I have already made a small presentation so that it will refresh your memories, whatever we have discussed in these module from module 5 to 8. And if you have any specific questions, you can ask me related to these topics. Padmasri? So just to remind you that this live session is only for, for a limited time period. So if you have any questions, you can ask me and take this opportunity to clarify your doubts, because it is very important for you to clarify your doubt to understand the forthcoming topics. You can type your queries in the chat box also, and Ramesh, do you have any question? If you don't have any question, I think we will wind up this session, and we'll wind up close sooner than what we are supposed to. Okay, so I think we have one question. Let me see the chat box. So Ramesh is asking how to clone a gene if gene is running in reverse direction. Ramesh, it doesn't matter whether the gene is running in a positive direction or the negative direction, because it hardly matters, because when you are doing a PCR, okay, you are actually going to you are going to use the site specific primers. So when you use the site specific primers, it will actually going to give you the amplified gene, and it will give you the amplified gene in the right orientation, and that's why it is not an issue actually. Even if it is running in the 5' 3' direction or 3' to 5' direction, it hardly matters, because you are going to use the primers accordingly. Then we have another question from the induction. So Srushti is asking what is mean by induction. So induction is that you are provoking, or induction is a English term is that you're provoking the cell to produce something. Okay, this means in this case we are actually provoking the cell with the molecule and asking that you should produce the protein of my interest. How it happens? It happens because the promoter. Now let me share my screen and I will explain you what is how the induction is actually going to work. Okay. Yes. So if you see the screen, okay. So see in a typical vector, right? So how the induction work is that, so induction means you are provoking the cell to produce protein or whatever the molecule you want. Okay, it means suppose for example I can provoke you for for beating someone, or I can do something, right? So if I do some action, you may actually be get provoked and you will do the action accordingly. So in this case what happened is that in a plasmid what you have is you have the promoter, right? So this is the promoter, and in this control of this promoter you have actually put your gene, right? So this is the gene what you have, right, which is actually you want the induction. So this promoter is actually binding the transcription factor. Okay, this is I'm telling you the general term. Okay, so transcription factor. Transcription factors are also proteins. So what you're going to do is you're going to put another plasmid, or you are actually going to put the genes within the genome of that particular organism as well. So in this one, what you're going to do is you're going to put the promoter and you're going to put this transcription factor gene under this control. So this promoter is actually going to get induced when you are actually adding the inducer. For example, if you take the promoter which is responding to the lactose, right? Then it is actually going to be induced by a lactose analog, which is called as IPTG. So if you use the IPTG, it is actually going to induce the promoter of the lactose gene, okay? And instead of gene, you have already put the transcription factor. So once the transcription factors are produced from this particular vector, it will be available, and that's how they are actually going to start transcription of the gene of your interest. Now, the next query is that we are, we have another question actually from the student, right? That is, can you, so I think it is clear to you, right, Srishti? Srishti, is it clear now?
Yes.
Srishti?
Yes, sir.
Clear, right? What do you mean by induction?
Yeah. Yeah, sir. Yeah, sir. Thank you, sir.
Okay. Yes. So then Subhadra is asking, can you explain the phenomena of salting and salting out? Okay. So again, I am sharing my screen and I'll show you what is mean by the salting in and salting out. Okay. So salting in is so before we go for this salting in and salting out thing, we have to first understand the protein structure. So if in a typical protein, what you have is you have a central core where you have the hydrophobic amino acid. Because hydrophobic amino acid does not like the water, so they are actually going to be in the central of this protein. And then this protein is actually going to have the hydrophilic amino acid, which is outside. Okay. Now, outside these outside this protein, okay, and then you also have the small hydrophobic patches also present on the protein. Okay, these patches are actually problematic because they may actually destabilize the protein structure. So to cover these patches, we also have multiple layers of hydration shell. Okay, so we have multiple layers of hydration shell outside this particular outer layer. Okay, this hydration shell is nothing but the water layer. So this is called as hydration shell. Now, what is happening is that if you add the salt, for example, so if you add the salt into this, okay, so what will happen is that salt is having the more solubility for the water. So salt has more affinity for water compared to the protein. So in that case, what happen is that the amount of the hydration shell, the amount of layers in the hydration shell is actually going to reduce. So after you add the small amount of salt, so salt you can add in a small amount, the protein is actually going to lose some of the hydration shells. Okay, like this. Okay, so imagine that you have 10 hydration shells here, so 10 layers of hydration shell, right? Here we may have maybe like 3. Now, because you have 3 hydration shell in this one, the diameter of this protein is low or less. Sorry, it's less compared to the diameter what you have here. Okay. So just for sake of understanding, suppose the diameter of this protein with the hydration shell is 10 nanometer, and if this is a 3 nanometer, this means you have actually compressed or you have reduced the volume what the protein is going to occupy. If that is the case, you can actually be able to add more amount of protein molecules into this. You can be able to add more protein molecule and they will be also get solubilized. And this phenomena is called the increase in solubility. Okay, because if you go by the definition of the solubility means concentration of solute or amount of solute, right? Which means the salt, right? Divided by the amount of water or amount of solvent. So amount of solvent remains the same, right? Here also I have taken 10 ml of the protein solution. Here also I have 10 ml of protein solution. Because you have removed some of the hydration shells, the diameter is less, right? So that's why the size of the protein molecule is less. So you have very lot of empty space, okay? And that empty space can be taken up by the additional protein molecule. That means you have actually increased the solubility. And this phenomena of adding the salt and increasing the solubility is called as salting in, which means with the help of the salt, you are inserting more molecules of protein. Means salting in, salt in, okay? So with the help of the salt, you are adding more amount of protein. Now, if you add more salt, even when the hydration shells are being removed, so what will happen? We are actually going to have this situation, okay? What situation we are going to have? You are going to have protein and you are also going to have so all the hydration shells are now gone, okay? This means you are going to have the patches which are not being covered by the hydration shells. In that case, what will happen is the neighboring protein molecules will also have the same patches, right? So this is the neighboring protein molecules, which are also going to have the patches. And these patches are hydrophobic patches. So they will actually going to form a bond. And imagine that if all the protein molecules are actually going to form the bond like this, they will actually going to form they will become insoluble. So they will not be going to soluble in water, or they are not going to soluble in water. So this phenomena of reducing the protein solubility, okay, so it will actually going to reduce the protein solubility, right? And this phenomena of reducing the protein solubility is called as salting out, which means with the help of the salt, you are removing the protein molecules. So you're removing the protein molecules from the solution. So earlier we were adding the protein molecule, that's why it is called salting in. Now we are adding more salt, right? So if you add more salt, you are actually exposing the hydrophobic patches, and that's how you are actually going to have the insoluble protein molecules. And that means you are actually reducing the protein solubility. That means this phenomenon is called as salting out. Is it clear now?
Yes, sir. Yes, sir. It's clear
Okay,
now. Sir, I
do you
have
have
another
any other
question.
question?
Can I? Yes, sir.
Yes.
Sir, according to the assignment 7, uh, the first question is thin layer chromatography is an example of. Sir, won't it be partition chromatography?
What kind of partition you see in a thin layer chromatography?
So, the solvent, the retardation factor is calculated, I think, and we calculate the distance traveled by the solute from the baseline and the solvent according to with respect to the solvent. So the correct answer is given as adsorption, but how is it adsorption chromatography?
Because the molecules are getting adsorbed onto the TLC plate. That's how they are getting separated. Adsorption chromatography means where the exchange of material will take place with the matrix. So all the chromatography techniques are adsorption chromatography, except the partition chromatography is where the molecules are going to be partitioned between the two phases.
Okay.
So here you don't have two phases. You have only one phase, that is the matrix phase.
Achha. Okay. Okay, sir. I'll just check. Okay, sir. Thank you.
Okay, very good. So anybody else? Anupriya, you have any question?
No, sir, no.
Okay, fine, good. Andrea? Ramesh, do you have any more question which you want to discuss or anything else? Not the question, but any aspect? Do you have any query related to forthcoming exam? Are you people sitting for the exam now? You have taken
Yes,
all
sir.
you have registered for the exam, right?
Yes, sir.
Good. Okay. So I think you know that the exam is going to be objective type questions, right? Where you're going to have 100 questions, and for every question you're going to have the 4 objectives. This time I have tried to cover up the complete content, and the questions are more or less going to be the same as what you have attempted already in the assignments. So they will be easy. They will be not out of the course syllabus, okay? And there's no negative marking. So you can attempt all the 100 questions because I'm sure you know the passing criteria of getting the certificate. And this time, in fact, I have already said to the NPTEL that if any student who would be interested to do internship in my course, internship in my lab, I'll be ready to take those students also. So basically, NPTEL normally gives the opportunity to the master students to participate into the NPTEL sponsored internship program for all those, especially like the toppers of the course, and that is a very good opportunity to be in the some of the labs in IIT than other places, so that you will get a better exposure. And on the top, NPTEL is going to provide you a fellowship. So that is a very good program to enroll. And the only criteria is that you should participate into the, the full course. You take the full course, you may be the, topper of that batch or something. Then only, or, at least you should qualify. So I think I will suggest to most of you that you should, participate into the exam and attempt all the questions because there's no negative marking. So, and then there's a high likely chances that you will get a certificate, and then you can also explore the opportunity of doing an internship in labs also. So, if Lokesh, do you have any question?
So no, sir.
Okay, fine. So, what is your background, Lokesh?
I'm a biotechnology UG final year.
Where? Where you are doing
University, Trivedi.
Huh?
University, Trivedi.
No, I can't hear you.
University, Trivedi.
Okay, fine. So, you're a PhD student.
BTech, sir.
So, first year student or the senior level student? So, if you don't have any more questions, I think we should wind up the session now. And and if you have any more queries in next week or some other time, you can easily drop your question in the discussion forum, and I'll be happy to answer. Every day morning, when I wake up, I normally try to answer all the questions what you post into the discussion forum. So that is also a very good place for interaction. And we can discuss all the questions. Okay. So if you don't have any more questions, maybe we should wind up this session. So I'm giving you 5 more minutes to post the questions, and then if I won't get anything in 5 minutes, we'll wind up the sessions. Okay, so I think it seems that you don't have any question, and whatever the questions we have discussed probably will help you to understand the topics. So with this, I would like to conclude today's live session, and I hope you will get benefited from this. And if you have any more questions, you can post that into the discussion forum, and I'll be happy to answer. Thank you.